Sean is the program manager for Microbiology and Grain Genomics at the Canadian Grain Commission, a department in the federal government of Canada. He's also an adjunct professor in the Department of Plant Science at the University of Manitoba. His program performs research on microbes such as fungi and bacteria on grain, as well as performs research for grain crop varietal identification. His research has been published in top-ranking journals, including Nature and Science. Sean has received numerous awards, including the Outstanding Young Scientist Award in 2022 from the Canadian Biotechnical Society. Today, Sean will present on the topic, Trends in Fusarium Head Blight in Canada. So Sean, it's yours.
Thanks, Shimash. All right, so hopefully you can see my screen okay. Sorry if you're staring at the side of my face. I'm sort of doing a dual screen here. Yeah, so I'm very excited to be talking to the EvoFunPath team of scientists and students, and to talk about Fusarium head blight, which I would say is maybe my second love next to my wife. I started working on Fusarium head blight as a graduate student, and in the early 2000s, I guess, so around 20 or so years ago. And then worked on it for my master's and my PhD, and then did a brief stint at the University of Saskatchewan working with the breeders, and then returned to the Grain Commission in 2019 to run the microbiology program. So I was able to return to working on Fusarium, which is wonderful. So one thing that I learned when I moved out west is how vast it is. Canada is just a massive country. It's a beautiful country for many different reasons, whether you're enjoying the sunrises over in Vancouver, when you can get them when it's not too cloudy, or the brilliant beaches on the East Coast. It's a really diverse country. And when you look at it, this entire area in the center of the country is massive. It's larger than many other countries in Europe, and this entire area is basically plains where we have massive agricultural production, just absolutely massive. So to give you an idea of how massive it is, these images are all sort of roughly to the same scale. And here you have Vancouver, and here you have Ottawa, where I grew up. I actually grew up sort of on the east end over here. And then here you have Swift Current. So you have, entire, fields that are thousands of acres for an individual section of land. And they're, the size of entire subdivisions in some of our largest cities in Canada. You could drive for a couple hours and you're just basically going through one town, Swift Current, that only has like a few thousand people. Like, it's absolutely mind-boggling how large these agricultural production is in some of these prairie locations in Western Canada. And if you look at, production of some of the grains in Canada, we produce over 30 million metric tons of wheat every year. And this is like, we almost have 30 million people in Canada. It'd be like if every person ate a metric ton of grain every year. It's just absolutely massive. And there's no way that any sort of individual could eat a metric ton of grain. So most of what we produce is exported to other countries. So we're a major exporter of high quality grain, and we need it to be really high quality so that the end users on the other end of the, that are buying Canadian grain are able to pay a premium for it so that we can ship it and go through all the, activities of exporting it to another country. So the Canadian Grain Commission, who I work for, we're sort of a regulatory body that sort of sets the standards for grain quality and safety. And we do a lot of monitoring within the industry, and we sort of set a lot of the rules and facilitate a lot of these grain transactions for exporting, and even domestically when, people are buying grain from producers and that sort of stuff. So we have sort of two divisions. One is the grain inspectors. They're sort of like the grain police. And then there's the research division, which we call the Grain Research Laboratory, and that's where I work. We have a total of 7 scientists working for the entire organization. So, you know, if you look at some other federal departments like Agriculture and Agri-Food Canada, they have hundreds of scientists. Like we have, 7, with myself being one. So the 7 programs are listed at the bottom there. My program is Microbiology and Grain Genomics, and we're part of this technology research group within the organization. So what we do on the research side is we do research on individual crops, looking at quality, diseases, and on the technology side, we look at, you know, chemistry-based methods for monitoring for pesticides and toxins that are produced by the microbes. My program does a lot on genetics and genomics, so we run a sequencing core facility, but we also have like all the tools for doing microbiology, like microscopes and incubators and all that sort of stuff. And a lot of what we do is monitoring of samples from harvest. And I'm going to really highlight some of our harvest monitoring in this presentation. But we also do monitoring of cargoes as well. We do a lot of requests for service analyses. So these are submitted samples because of weird things that happen in industry, or if there's some sort of market access issue or a trade dispute. They sort of, you know, call on the scientists to save the day, to provide science-based support to show that an organism is either there or not there in the grain. And we also evaluate lines that are going for registration for being grown by producers. One thing that's weird about us is our headquarters is actually in Winnipeg. Most of the federal government is located in Ottawa, but we're actually, most of the organization is here in Winnipeg, and I'm actually on the University of Manitoba campus, so I'm even a bigger weirdo when it comes to this sort of thing. So I'm sort of an island, but it's nice because I can work more closely with some of the people even on this call that are on campus, and we can host students and that sort of thing. So there's lots of different grains that are produced across Canada: barley, oat, rye, triticale, wheat on the cereal side. And that's where I'm going to sort of focus today, is on the cereals, mostly on wheat, because we're talking about Fusarium head blight, but there's lots of different other grains that we'll look at too. And on the microbiology side, we're looking for fungi and bacteria. So the fungi and bacteria could either be plant or animal pathogens, but they could also be beneficial microbes on the plant or in human and animals. And we sort of look at all of them. So on the fungal side, we do a lot of work on Fusarium and ergot. If you're not familiar with ergot, it's also a very cool fungus. It makes lysergic acid derivatives, which is LSD. It's a precursor to, you know, hallucinogens, which is the street drug acid. Anyways, very fascinating fungus. Maybe a second love to Fusarium, or third love, I guess. On the bacteria side, we look at a lot of different plant pathogens. Right now, there's a lot of work being done on the evolution of leaf streak pathogens and Xanthomonas. We also look at gloom rot, Pseudomonas, and some of these other bacteria. On the human and animal pathogen side, we do a lot of work on E. coli, Salmonella, Listeria. But it's rare for these to actually end up in end products to result in foodborne illness, but there have been cases of product recalls due to the occurrence of some of these bacteria in products. So shown here are some of the technicians within the biosafety cabinets, and this is what, you know, Fusarium looks like on the grain. If you take grain that looks infected with Fusarium and throw it on some PEA, you'll get some colonies growing off of them that looks like this. These black ones are probably alternaria. So the Canadian Grain Commission, we're not sort of, you know, working in a bubble. We work with farmers, industry associations, grain handling companies, other federal departments to make sure that grain is fit for consumption. You know, we all have different roles and responsibilities in making sure that this happens. So we establish grading factors and inspect grain. That's mostly what we do. And we monitor for shipments for chemicals and biological things. And we do research of methods to be able to better detect these. Whereas, you know, Health Canada and Canadian Food Inspection Agency, they're more involved with setting limits for, how much toxins might be allowed in certain things, enforcing some of the rules and looking more at food. Agriculture and Agri-Food Canada, they do more agronomy and breeding and this type of stuff. And then sort of on the applied end, the farmers are doing a lot on their end to make sure that there isn't diseases on their farms. And the industry associations sort of help out the producers, give them guidance to better manage things on their farm, make sure that there's communication across the sector. And then, grain handling companies buy the grain from the producers, and they might need to clean the grain to remove certain things from it and this type of stuff. So we're all doing our part. One of the main things we do is inspections. And we have grain inspectors that actually go through and look at grain. We've been doing this for over, almost 100 years. You can tell by the age of this computer, maybe by how long we've been doing it. But this is actually part of an exhibit from the Manitoba Museum, and this is a certificate of inspection from 1930 from Churchill. So this is a grain shipment that maybe went to my grandfather over in Europe that Canada was shipping out, almost 100 years ago, and the Grain Commission was at the ports there inspecting the grain to make sure that it was containing the appropriate amount of, quality and was free from diseases and these types of things. So they'll actually go through and inspect the grain. So here, this is an ergot sclerotia. But, here we're going to talk about fusarium damage. So this is, varying degrees of fusarium damage. Usually you see these chalky white occurrence sort of on the outside of the grain. So fusarium head blight is caused by fungi from the fusarium species. And usually you see these leaking symptoms. So this would be, a healthy looking wheat head. And then these were ones that were point inoculated. And then you can see that they sort of turn white. Sometimes you get these phenolic loading of brown appearing on some of the tissues. And, some of the kernels may be aborted. They might become covered in fungi, become shrunken and shriveled. And they often contain toxins that the fungi are producing. And one of the ones that's most concerning is this toxin here called deoxynivalenol. It's also called vomit toxin. So the fungus itself, it's an ascomycete. It overwinters in its sexual structures. The paracecia, the spores are discharged, and it's timed almost with the heading of the plant, so that the spores can land on the plant just as anthesis is starting, just when the plant's starting to flower. And then the sexual spores will land on the plant, initiate infection, start colonizing the grain, and then it'll start to colonize the tissues and produce asexual spores that can infect neighboring plants and then sort of spread throughout a field. So these sexual spores can actually be discharged quite far. I think it showed that one of the fastest accelerations of any living thing is actually a fungal spore from a sexual structure from the fungus. So, these things get discharged quite far. They'll land on the grain, they infect, the asexual spores get made, and they can splash around to other plants. Once the plant dies, the fungus continues to eat the dead tissues, and then it starts to make sexual structures and begins to overwinter. So because of its life cycle, it's a bit of it's not a biotroph, so it doesn't live sort of as in a parasitic relationship where it's keeping the host alive. It actually kills the host and then will eat the dead tissue. So sometimes people call this a hemibiotroph. So it starts off sort of living in this pseudo-biotrophic way, and then it kills it, and then it eats the dead stuff too. So it occupies sort of two different lifestyles almost, depending on where you're looking at in the infection stage of the plant. And then again, the problem with this plant disease is, in addition to causing the yield losses, these grains will be contaminated with these toxins, and that's a problem because the toxins themselves are regulated. So this disease wasn't always a problem in Canada. It really started picking up in the 1990s, but even if you look back to the 1980s, There was some work done by the Grain Commission and others looking at the different species that were causing this disease. So this is a table from a paper that came out in 1990 from Randy Clear, who is my predecessor's predecessor. And, they're looking at the different Fusarium species that cause the disease. And you can see that, the disease wasn't really prevalent in red spring wheat or winter wheats, but there's a lot more of it in durum wheat, which is a different species. It's used for making pasta and couscous, whereas spring wheat and winter wheat are used for making bread and noodles. But we did see, Fusarium graminearum in a lot of the kernels, but you also saw a lot of these other species, Fusarium sporotrichoides and Avanesium and acuminatum. Fusarium, this is by kernel, this is another table by sample. So they looked at like a 500-gram sample, and then they said, were they positive for the different species? And most of the samples for durum were positive for sporotrichoides, and Poe and Avanesium, and then graminearum was sort of lesser of disease, especially in Western Canada. In Ontario, there was a little bit more graminearum going on, but there were a lot of different species causing the disease in the 1980s. And this is another, I found this gem in an unpublished report. It was a banker box hidden in an office desk somewhere. And it's a map that somebody drew in the 1980s. And they were looking at this Fusarium graminearum species. And it looks like, they started detecting it in southern Manitoba in 1980s, early 1980s. And then it started to spread throughout South Manitoba. And then in 1990s, these are all newspaper clippings from 1993. It just blew up. The disease just exploded in Manitoba. So there are all these articles, even from the popular media, of this toxic harvest from Fusarium infecting different grains in Canada. In some samples, 38% of the kernels of this one variety had this disease in it. So there were all these stories that were coming back from individual farmers that showed that this disease was just exploding in Manitoba especially. And this sort of brings us more to the work that I've been working on. So at the Grain Commission, we have something called the Harvest Sample Program, and this is a voluntary program that producers participate in. It's started in 1995, where we're receiving producer samples. They basically take grain, they throw it in an envelope, dump it in a Canada Post bin, and then it comes to our headquarters, and then we assess it and give the producer a preliminary or unofficial grade, and that helps them sell their grain because they know roughly what the quality is. But we can actually look at that grain and look for all the grading factors, including Fusarium. And we've done this, since 1995 for over 300,000 wheat samples. So this is like a massive, massive data set. And this is what the data shows. So early 1990s, like 1995, not a whole lot going on in terms of Fusarium across the different wheat classes. So this is winter, this is durum, this is red spring. Red spring is the most popular wheat that's grown in Canada, this red line. And you can see that it just took off in the 90s, and then it sort of peaked here in 2016. In 2016, it was so bad, they estimated that there was $1 billion in economic losses due to Fusarium in 2016. Like, it was just unbelievable. And it was a little bit different from winter wheat, and you can probably imagine why from that life cycle image that I showed you before. The disperse of the spores is really linked to heading. In winter wheats, they head a lot earlier. So there's sort of this asynchronous amount of inoculum that was appearing on the spring wheats versus the winter wheats. But anyways, 2016 was terrible. You can see that Manitoba was a lot worse from the beginning, probably because we had some more aggressive species popping up, like Fusarium graminearum. But in 2016 and some of these other years, like it was bad across all of the different crop types. And if you look here, we don't have any data really going past 2006 in Manitoba for durum wheat, because farmers stopped growing durum wheat in Manitoba. And one of the main reasons why they stopped growing durum wheat was because of how much fusarium was there. It was so bad that they just didn't even grow the crop there anymore. So they started growing durum in these areas that were much drier and less prone to having disease. Here's the data that we've sort of broken up even further by crop district. So you could take these Western prairie provinces where we have that sort of bubble where we grow most of the wheat, and you sort of break it up more by region, and you can see how bad the incidence of fusarium in Manitoba was across all the crop districts. And, you know, it was universal. And then you can see in Saskatchewan, it gradually picked up, but it was across all the crop districts. Likewise for Alberta, it's still sort of picking up, but Alberta and parts of southwestern Saskatchewan, like 4A and 4B, where you can see here, there's less fusarium because it's a little bit drier. There's less moisture, so it discourages the growth of fungus. And then we see the levels of severity here. So this is, you know, the number of samples that would have fusarium, and this is if they had fusarium, how bad was it? And you can see that it actually gets a little bit better in Manitoba, which is kind of interesting. Maybe farmers are doing something different to manage the disease on their farms, which is reducing the severity of the disease on their farms. So things like applying fungicides, growing disease-resistant cultivars, rotating crops, and stuff. So here's the same data, but I've done it sort of as a time series where you can see it as an animation. So you can see the disease is slow going into the 2000s and is slowly picking up for both red spring and durum wheat. And then you can see once we hit 2016, it's just like a completely just nasty year. So you can see just how bad it was in 2016 for incidence. So, you know, the disease varies by location and year. And in 2016 was a bad year. So what's affecting these levels of fusarium? And for this, I'm going to go back to the old school disease triangle, where we have this interaction between host, pathogen, and environment. And if you have sort of the positive sort of interaction between all these variables, it can result in disease. And I think, you know, there's this classic way of thinking about host, pathogen, and environment. And from an evolutionary standpoint, many of us often go straight to cultivar resistance, effectors, and environment being things like moisture. Especially, you know, being a geneticist myself, this is sort of the mindset that I often go to. But I'm challenging the people on this call not to always just think about these things as being factors that control this type of interaction. These interactions are actually much more complex. So on the host side, you know, you have cultivar resistance, which might be genetic, but you have all these physiological things that are happening as well. You have things like the flowering time, the height of the plant, you know, what the species is. We're clearly seeing a difference between durum wheat and red wheat, for example. You know, how well the anthers open up to make the flowers more willing to receive spores to initiate disease. So these interactions are actually quite complex. On the pathogen side, what the inoculum levels are, what the species and strains are, what types of toxins they're making, or what the chemotypes are. If they have resistance to the fungicides, if they have, you know, tolerances to different types of climates, if they prefer drier conditions or wet conditions, depending on the species or strain, all of these things will be affecting, you know, driving the evolution of the pathogen and, you know, the success of this pathogen system as a disease. On the environment side, we also have, you know, all the effects of climate change. We have temperature, we have moisture, but we also have, you know, what type of soil it is, how that's affecting the health of the plant, you know, and you know, the other microbes that are appearing on the grain as well, including other fusarium species. And I'm going to talk a little bit about that as well. The interaction between, you know, individual isolates and different species. So a lot of the early work that was done to try to characterize the diversity and the evolution of the different species was done using sort of old school classical microbiology. And I think that this is actually a bit of a lost art. More people should be doing some of this classical microbiology. So if you do have genetics and genomics components to your projects, I strongly encourage you to, take a picture of this thing under the microscope, because not enough people are doing that anymore. But anyways, so you could actually tell the different species apart based on the colony morphologies as well as the spores. So for example, at the top here, this is Fusarium graminearum. It has a little foot at the end of the spore, and it's like highly septated. This is Fusarium culmorum. It's a little bit of a shorter, fatter spore. This one here is Trisynctum. It's a very skinny spore. So the way that, old school mycologists would identify these different species and characterize their diversity is by inspecting them under the microscope. But genomics really changed a lot of the way that we look at evolution and of these fungi. So of course, DNA gives rise to RNA, which codes proteins, and these are the functional units within cells. And this is what's allowing the fungus to presumably have different degrees of aggressiveness or to behave differently than each other. And the cost of sequencing DNA to understand these differences has gone down, astronomically. Even when the human genome was first sequenced, there was maybe $100 million or so, but now you can get a genome for around $100. So that's done routinely in any lab. People can buy a MinION sequencer for $1,000, and like off you go, you're sequencing. Interestingly, the Fusarium genome was actually one of the first fungal genomes to be sequenced. It was sequenced using some shotgun Sanger sequencing way back in the day, using the same approaches that were used to do the first human genome, because of how important the disease was at the time, picking up in the 90s. So the sequence was completed in 2002. And this actually, combined with the importance of the disease, resulted in a massive influx of research productivity in Fusarium and trying to understand the biology and evolution of the pathogen. If you look at tourists that have Fusarium graminearum in the title for NCBI, there's relatively few. The genome came out in 2002, and then the amount of research going into trying to understand this bug is just gone up massively. A lot of the work was done by Canadians too. So this is a blast from the past. So this is a picture of me during my PhD days. My supervisor was Gopal Subramaniam, who's a part of the EvoFunPath team. And I think the Chris is also part of EvoFunPath. He's now doing his PhD, but he was an undergraduate student at the time. And there's other scientists like Liz Brower, who's now at AFC. They're continuing some of the phenomics work on wheat and other aspects of Fusarium as well. So we looked at, trying to characterize some of the genes involved in making sexual structures and making toxins and regulating toxins and factors involved with aggressiveness of the types. So some of the early research done, not just by us, but others as well, identified the genes responsible for making and regulating the production of toxins, including deoxynivalenol. And they're actually all co-located in a cluster. And there's some key genes like tri-8, for example. There's a allelic variation in this gene that can give rise to different forms of the toxin. And then there's another gene located outside of the cluster, tri-1, and allelic variation gives rise to this other toxin variant called NX2. So I'm going to talk a little bit more about 15-adon and 3-adon, these two different toxin variants that are the result of allelic variation in this gene tri-8. And there's also in parallel all this work that's going on for other species too, and we can actually develop markers that are specific for tracking the different chemotypes, the 15-adon and the 3-adon, as well as tracking all the different species as well. So these DNA tools became available, and we can now use, instead of using classical microbiology, we can use high throughput DNA testing to be able to understand these organisms a little bit better and monitor for them better. So this is an image from my lab. It's this is the sequencing lab. So we've got some Illumina sequencers, some nanopore sequencers, and we've got the PacBio sequencer here. And we also have some high throughput phenotyping equipment. So these are like doing PCR but instead of running it in strip tubes where you have 8 or 12 or 96 wells, this is a 5,200 well system, and it's all fed through with robotics. This is a video of what that robotic system looks like. We have our DNA samples that we get from individual kernels, and we put the DNA in these 384 well plates in the back here. And then this machine here grabs the DNA sample and then pipettes them here. Each one of those little holes in the chip holds 100 nanoliters. So they're 100 nanoliter reactions. And we'll run 24 different DNA tests on each kernel using that table that I showed you before with all the markers. And based on the pattern of those DNA tests, we can identify what the species is, what sort of virulence factors or effectors they might have, what toxin genes they might have. And then this is sort of the outcome of that analysis. So here we have some recent data on the different Fusarium species across from 2014 to 2020. And you can see that, the most dominant species is Fusarium graminearum. So, in some of those early 90s and 80s studies, I was showing you, Avanasium and Acuminatum and Sporotrichoides were quite common. Now, across the board, we're pretty much seeing Fusarium graminearum as the Fusarium king when it comes to Fusarium head blight. What's interesting is Alberta, which is a little bit drier, you actually still see quite a bit of Fusarium avanasium on the grain. And that's important for us to understand because Fusarium avanasium doesn't make deoxynivalenol or DON. It doesn't have the genes required to be able to do that. So there is still quite a bit of diversity out there in the field, both in terms of the different types of species that are occurring. And you can see the sample numbers for this are absolutely massive. The table that I was showing you from the 80s, it's somebody staring through a microscope and growing the culture. It's really laborious. It takes a long time. It's a lot of work to be able to do that. Here we're testing, in 2014, we tested 8,611 kernels. These are just absolutely massive data sets because you can automate all the DNA isolation and the DNA testing. So we can run these massive surveys and really get an idea of what's going on. So if you look across Alberta, the diversity is seen sort of across all the different crop districts. Saskatchewan, you're looking at a little bit more diversity, but less so than Alberta. You're still dominated by Fusarium graminearum. And then here in Manitoba, we're almost seeing, in some years, it's almost exclusively Fusarium graminearum, like in this 2016 year, which was that huge epidemic with the $1 billion in losses. That was a heavy Fusarium graminearum year, both in Manitoba and Saskatchewan. But you can see Alberta was still quite diverse. We could also look at the different toxins that are being made using those toxin-specific DNA markers. So here at the top, we have the marker that we used for assessing 15A-DON. And you can see that even across our sample years, 2014 to 2020, there's a decrease in the amount of 15A-DON genotypes that we observed. In Alberta, Saskatchewan, Manitoba is a bit level. And in Eastern Canada, there's a little bit of a reduction as well. And if you look at the 3A-DON, which is a separate marker, when 15A-DON disappeared, 3A-DON went up to take its place. So we're seeing an increase in the 3A-DON producing isolates in Alberta, Saskatchewan. And this is even, just over 6 years. You can see that the population structure of the fungus is still changing, which is really, really interesting. This is hexaploid wheat, but the results are even a little bit maybe more pronounced in durum wheat, especially in Alberta, and Saskatchewan maybe it's a little bit more level. And again, we don't really have durum wheat data for Manitoba because no one grows durum wheat in Manitoba anymore, really, because of the amount of Fusarium that's there and how much more susceptible durum wheat is to Fusarium than bread wheat. So we could also look at the amount, the severity of those samples for Fusarium and the amount of DON toxin that's there. So we assessed every sample that goes through the harvest sample program. If it's wheat, it also gets assessed for the amount of toxins being present. So this is like a really well-oiled machine. We receive about 10 to 15,000 samples a year. So each are individually inspected. assessed for dawn. And you can see that the severity is sort of a large range in terms of severities that we observe, but generally durum wheat is a little bit more severe than bread wheat is. And then this correlates with an increased level of this toxin that's appearing in the grain as well. So, we're continuing to do research in the fungus trend to find new ways to identify the fungi and characterize their diversity. So Shimosh, who introduced me, is a graduate student in the lab. So he has this project using mass spectrometry and biochemical profiling to be able to identify the different species, and it seems to be working quite well. I'm not going to present on that because I'm sure you'll get a talk from him at some point in the EvoFunPath group, but I won't steal his thunder. But this is a really exciting project. His results are really looking good. It was funded through different producer groups. But to give you an idea of even what you can get in terms of complex interactions, like these, we have two different species here, Fusarium graminearum, or strains, FG1 and FG2. So FG1 infects quite a bit more than FG2, and you can see that when you dilute it out with water, you're not seeing that much of an effect on disease. But when you start combining the two fungi together, you actually see a marked reduction in disease. So this has this sample here, for example, has the same level of inoculum of FG1 and FG2, but you're seeing, almost a 20% reduction in the amount of disease. And this is causing a reduction in the amount of deoxynivalenol appearing as well. So when you inoculate even the same species together, they're interacting with each other, and this is causing interference that's reducing the amount of disease that's appearing on the grain. So this, I guess, is a word of caution. We often do these really controlled experiments in greenhouses and growth cabinets, but in the field, we have these really complex interactions between different species of this Fusarium, as well as Fusarium and other microbes that might be on the grain, as well as within the species. Even when we combine the isolates within species, we're getting these complex interactions. So in the field, you often get these very different results than you get in the greenhouse or the growth cabinets. And it's because of all these complex interactions that are going on. Again, I talked a little bit about the pathogen. I'm going to be very brief and just talk maybe for 2 minutes on host and environment, because they're really an important part of understanding the evolution of the pathogen. So some of the work that we've been doing is characterizing the diversity of the host. So I was part of the pan genome for wheat, where we sequenced diverse wheat from around the world and characterized their diversity. One of the things that we looked at were the disease-resistant genes. So these are NLRs, so nucleotide binding leucine-rich repeat genes. So these are genes that would typically be receptors for identifying fungi and initiating defense response signaling. And, I guess model systems like Brachypodium or Arabidopsis, you have a few hundred of these genes. In wheat, there's massive gene expansion. It's unbelievable. You start off with around 2,000 of these genes in a single genome. And once you look at their diversity across different cultivars, you're looking at, 8,000 different NLRs. So trying to understand how these receptors are interacting with each other becomes very, very difficult when you have a really large, complex genome like this. And it gets even more complex when you start to factor in the diversity that's available within the host across different cultivars that are being grown. But you can start looking at, the differences at the genetic level. You can look for, different mutations and the effects that these would have on protein function to identify new alleles and try to predict which genes might be important for improving the crop or interacting with the pathogen. And this can be done both on the pathogen side and the crop side to sort of understand what the effectors are and what the receptors are that are sort of responding to it. So we've been doing some work when we find genes, even with complete presence-absence variation, that we think are probably involved in susceptibility. So they're interacting with pathogen effectors. And the presence of these genes is causing the pathogen sort of hijacks them and uses them as a way to make the plant susceptible. So if you lose these genes, we have some evidence to say that it's giving rise to more disease resistance. But these genes might be important for resistance to another pathogen or something like this. And this is where the interactions become complex. On the environment side, this is where it gets, once you're packing on all these different types of data, it becomes difficult to sort of parse what's important and what isn't. And this is where machine learning and AI becomes really, really important. So we've been applying machine learning and deep learning to our microbes and microbiome data on different wheat crops, combining this with environment data and crop phenotype data and our genetic and genomics data and these models to be able to predict different things. So to give you an idea of what this looks like, you have your different inputs, and these might be station data for different weather stations across the different crop districts, and what the temperature and humidity was, or how much rain went, or even satellite data. So each one of these pixels is 9 square kilometers, and we can sort of aggregate data for precipitation and humidity by different regions. We can combine it with soil type data, and we've written some code to put them automatically pull all this data with the fusarium data, put it into models, and see and do different types of predictions. And this is part of a project that we have going that was just announced with Genome Prairie and Genome Canada to look at climate smart microbes and microbiomes, and to sort of do all these sort of genomics in the crop side, microbe and environment interactions. And we're trying to really get at some of these different questions, like what the relationship is between the environment, geography, soil type, weather, and how does this affect FHB incidence and severity, disease resistance, and shifts in these pathogen populations that we're observing. And, are there any varieties that are performing better or worse than expected in certain areas? And maybe this can tell us, there's a specific genetics in that pathogen in that area, or maybe the cultivar in that area that's giving rise to reduced disease, and we can sort of take that and use it to our advantage to better manage the disease. Can we improve forecasting tools, predict fusarium outbreaks before they happen using weather information? This way farmers can, use tools and like fungicides to, better manage the disease on their farm or time them more appropriately, so that they know that, this is going to be a bad year for fusarium. I know that, I need to be a little bit more aggressive in my management, or, what, this year's actually not too bad. I need to worry about other things. And, that does happen too. Like in 2021, we had a drought across the prairies, and there's hardly any fusarium because there's just no moisture. What was interesting though is the fusarium that we did observe, a lot of it wasn't Fusarium graminearum, it was the other species. Thereby, also supporting that there's this complex interaction between environment and the plant pathogen populations as well. And also by looking at areas that see more or less fusarium damage than expected, we can talk to the farmers and producer groups and sort of better understand what they're doing in those areas to manage the disease. Maybe there's tricks or something like that they're doing, and we can share those practices across the industry. So I'm going to end the presentation there, but with some take-home messages. One is, Canada is a major producer and exporter of grains. We are, a powerhouse in agriculture, in horticulture and other things as well. But, until you drive across the prairies and see how massive those fields are, like, it's unbelievable how much we actually produce in Western Canada in agriculture. These grains, they encounter disease pressures from fungi and other diseases. And these are caused by complex interacting factors. We talked about the host, the environment, and the pathogen. And the pathogen's always evolving, but this is also, evolving in tune with environment and the host. So it's important to look at all of them. And don't be fixated on, gene for gene interactions or like effectors and, receptors or just moisture. There's all of these other factors that are in play to manage those interactions. So think big. And, sort of to end on a positive note, despite all the challenges that we have in managing some of these diseases, there's a lot of checks and balances in place. So the Canadian Grain Commission We have a robust quality and safety assurance system for inspecting the grain, grading it, performing research, and this, makes sure that the grain that we export from Canada is always, the top-notch stuff, because it needs to be, because we are an exporting nation, so we are depending on other people that are buying Canadian grains to, be confident that the grain is meeting their needs. And I'd like to acknowledge the other people in the program that do the work. I'm just sort of the face that gives the presentations, but it's all the hard people in the lab that actually do the work, especially the grain inspectors and the people that are managing our harvest sample program. And the producers that grow the grain and send us the sample, like they're probably working harder than any of us here at the CGC, I'd argue. But anyways, and here's the team. We've got Shimash, myself, and we have some biologists and technicians that come along. And this is us at a field tour out in Brandon at their breeding facility. Yeah, and I'm happy to answer any of your questions.